Hello everyone. Is my screen visible? Okay. Okay, great. Okay. So, good evening and welcome to this course, a certificate course on radiocarbon dating, or as you all may call it, the C14 dating in archaeology. Now, this is one of the most powerful scientific tools in archaeology, and it's still a dating method that many archaeologists rely on till date. So I'll briefly introduce you with this topic, and I hope that it's going to be interesting for all of us. So now let's begin. So the very first question, like I'll begin with a very fundamental question: why does dating matter at all in archaeology? So now think that you're all at some site, any site of your choice. For example, Tavira, and wait a second. Okay, okay, okay. So yes, so I was talking about imagining any site in your mind, and you find a piece of charcoal over there, along with some potsherds. Being an archaeologist, you already know that this material from a particular layer that you found is older than any material you found from a layer above it. Now, this is relative dating. And how do you know that this was the layer that you found your material from is older than the other layer which was above it? By stratigraphy, the law of superposition. Now, you may also look at the potsherds and claim that this is new as compared to the other type of potsherds from the same site, maybe layers below that. Now, this would be typology and seriation. Further, we have cross dating with the known assemblages as well. Now, the thing is, all of these methods, they will tell you the sequence. They will not tell you the exact age. They will not tell you the exact, they will not answer the exact question of when. Now, think about how limiting that is. For example, if we had relative dating, like only relative dating, even now, we could say that the Harappan civilization came before the PGW period or painted graveware culture. But we could not tell by how many centuries or whether they overlapped or whether this transition happened quickly or what was the what were the whereabouts of these. So that would be a limitation. That is the exact question that matters in archaeology and that matters for understanding history. To solve this, chronometric dating came into being. Now, with this, we get absolute dates. These methods, these chronometric dating methods, they're based on measurable physical and chemical processes. These include processes like radiocarbon dating, thermoluminescence dating, OSL dating, dendrochronology, etc. Now, these dates, these methods, they do not provide you with any guesses. Like they don't give you guesses, they guess answers. They give you dates, the actual numeric dates. For example, 3000 BCE, 200 CE, etc. They are true measurements that you get with precise science. Now, why does it all even matter? Now, we need all of this to reconstruct the sequence of human events. We also need this to test the historical hypothesis. Now, these hypotheses, while we are testing them, these might come out to be true, but we need evidences to provide that. We need empirical evidence to kind of test those hypotheses that we have historically or by scholars. Then it also helps us in understanding the cultural change over time, not only in one country but also for comparative studies. So it helps us tell how the society wherein we have no written records. To get the dates for those particular sites with precision, we need chronometric dating. So, in that way, chronometric dating has been very important in the field of archaeology. Now, coming to the method, sorry, yes, so now coming to this method, wait. Uh, A second. My screen is still visible, right? Yes. Okay. So now we'll come to radiocarbon dating. So if we talk about it is visible, right? Okay, okay. Thank you. So now coming to radiocarbon dating, which is the topic of this lecture today. So this method was developed by this person, uh, W.F. Libby. Now you guys must have, I guess you guys must have heard of this name before. Now he was not an archaeologist; he was a chemist. So he worked at University of Chicago, and he was working on natural radioactivity in environment. So now it is not that he discovered C14 or carbon 14. Scholars before him, like scholars in 1930s, were actively working on carbon 14. They had discovered and noted the presence of carbon 14 in the atmosphere. But he proposed radiocarbon dating. So, what he observed was that every living organism continuously exchanges carbon with the atmosphere. They do this by breathing or by photosynthesis or by eating, like living organisms, plants, and animals, humans, everyone. Is okay, the screen is not visible. Wait, it okay, okay. So, and they do this by they do this by all of these processes, and they do it for as long as the organism is alive. So for as long as we are alive, we are in that cycle of converting carbon 14. So and we will do it till we die. Okay. So he also said that now this carbon 14 maintains the same ratio as the carbon 12 in atmosphere. We'll be talking about this in the further slides in more detail. So this is I'm kind of just trying to explain the his work, like his research on this topic. So, so he is the one who came out, who proposed this research that when an organism dies and this exchange stops, and the carbon 14 that was there already present in this organism, it starts decaying at a known rate. This measurement, like if we measure that rate, if we measure that remaining carbon 14 and the rate by which it decays, we can actually go back to how long it has been since the organism died. Now, to prove his work, he first tested this method against the Egyptian artifacts, whose historical dates were already known from the written records. And what came out to be the result was that these two dates, like his dates, his tested dates, and the historical records, the historical dates, they matched. And this is the thing that gave his research validation. This validation got instant credibility because of this particular test that he did on the Egyptian artifacts. And because of this work, he also won the Nobel Prize in Chemistry in 1960. Now, further, a big revolution came in this particular aspect of radiocarbon dating in 1977 when we got AMS dating as well. Now, we'll be talking about this later on. Now also, yes, when I'm talking about his testing of hypothesis, what makes it so important and so interesting is that he used a completely independent line of evidence, the historical records, to confirm a physics based measurement. This is basically a gold standard in science. When we have two different, like two entirely different, actually, entirely different methods arriving to the same answer, this means that you are correct, right? This gives us this confidence in our answer. It gives us this credibility. Now, that principle testing your dates against the independent evidence wherever possible is something every archaeologist should keep in mind. Now, India was not far behind when I'm talking about this. India also, in India, we had the one of the earliest radiocarbon dating laboratories of Asia, of the entire continent, which was opened by Tata Institute of Fundamental Research. It was first in Bombay. The lab was first in Bombay. However, it was later on shifted to Ahmedabad, and it is now known as Physical Research Laboratory, PRL. So this PRL laboratory, it operates under the Government of India, Department of Space, basically of the Government of India, and has been running these radiocarbon dates for Indian archaeological and geological sites since 1970s. They also developed their own AMS graphitization facility, which means that Indian researchers no longer required to rely on the foreign laboratories for precise measurements. Further, we also have BSIT, Birbal Sahani Institute of Paleosciences in Lucknow, where we have these facilities for AMS and radiocarbon dating. For AMS, they also made their own sample preparation, got their own sample preparation machine in 2017 or 18. You'll have to check which year it was exactly. And these two institutes, I believe, are safe to be called the backbone of radiocarbon dating in India, at least. And then now, after introducing you guys a little with the history of radiocarbon dating, it is also important for us, like if you want to understand radiocarbon dating, it is also important for us to know a little bit about carbon chemistry, like not too much. I'm also not a scientist, I'm not a science student either, but then a little bit of an understanding of how this works, how carbon atoms are formed and etc. would help us in understanding radiocarbon dating as a whole process. So keeping it all very basic and introductory, easy to understand for all of us, we know that carbon is one of the most abundant elements in all living beings, right? But then it has different atoms, and not all atoms are identical. They come in different forms, which are called isotopes, which are chemically identical, but they differ in number of neutrons in their nucleus. So based on this, we have three over here: carbon 12, carbon 13, and carbon 14. Carbon 12 is the most common one, which makes up to about 98.9% of all carbon. Then we have carbon 13, C13, which makes up about 1% of the entire carbon, and the remaining, which is very tiny amount, very trace amount, which is carbon 14. Now these carbon 12 and carbon 13, both of these are stable, while C14, carbon 14, is radioactive, and this is what makes it special. This is why we have C14 for dating and not C12 or C13. Now, if we, like, while we are talking about this, we also need to know that the half-life of carbon 14 is 5730 years, which you all should remember. I suppose, like, you should remember. I'll recommend you to remember that date. And also, because we are talking about the formation of these isotopes, particularly about carbon 14, it is created in the upper atmosphere through a nuclear reaction. Okay, so what happens is the cosmic rays from the space, they travel down to the atmosphere and they create a secondary neutron. Okay, now this neutron collides with the nitrogen 14 isotope present in the atmosphere, the upper atmosphere, and these two they release one proton and also carbon 14, like this becomes carbon 14, C14. Now this freshly made carbon 14, it oxidizes into carbon dioxide, so it is basically called 14 carbon oxide. So carbon dioxide, so it becomes carbon dioxide and then it mixes with the atmosphere. So, okay, so I have a graph over here. Before that, we'll be also talking about different steps which are involved in this, like just just to elaborate a little bit on what I've told till now. So what happens over here with this carbon 14 is it oxidizes, and then while it is there in the atmosphere now as carbon dioxide, the plants absorb it through photosynthesis. The animals also eat these plants, and hence they also get this carbon 14 in their bodies. Then the carnivores who eat these plants, who eat these animals who eat those plants, also get the same carbon 14. So in that way, every living thing on this planet, including everyone over here in this meeting, you and me, we all have that carbon 14 cycling through our bodies right now, and it is also maintaining the same ratio as is there in the environment. So to give you an idea, in every single atom of carbon 14, we have 1 trillion carbon 12 atoms. So that is the ratio in the atmosphere. Now telling you a little bit about, like, elaborating on these steps which I've told over here. So the first step over here is carbon 14 production. So as I said, the cosmic rays, they're bombarded in the atmosphere, and they react with the nitrogen 14 present in the atmosphere, producing carbon 14 atoms. Now this is a constant process and it keeps on happening. Then we have atmospheric mixing. So this carbon 14 atoms, they oxidizes to form carbon dioxide, which mixes throughout the atmosphere. Then they are observed by the plants, they are observed by the animals, all animals, herbivores and carnivores, and omnivores as well. Uh, and all of these living organisms, they maintain the atmospheric carbon 14 to carbon 12 ratio. Then this process, this cycling continues till the organism dies. Now, at death, which is very important, this is the most important aspect of this cycle. So, the moment an organism dies, whether it's a tree which fell down or an organism, an animal that died, this exchange of carbon, it stops in that particular organism. So, and no new carbon 14 enters the body. And now we already had some aspect of that carbon 14 because it was a continuous process. No new carbon is entering our body, but the carbon 14 that we already had in our body, it starts to decay now, and it starts to decay back into carbon 14, into nitrogen 14 that is there in the atmosphere. Now this process of decaying of carbon 14 into nitrogen 14 is called beta decay. And then comes the step of measurement. Now when we send this sample of, for example, charcoal to the lab, the remaining carbon 14 is measured using beta counting or by AMS. So we'll be talking about these two methods later on. And then the final stage of age calculation. So the answer or the result that we get by using the half life, which I told you, 5,730 years, it does not come in it does not come in calendar years. It comes in, like, it'll give you something in range of before present. So BP term will be used for this. Now this does not mean from 2026, it means from 1950. Yes. What is this screen not okay? It is, right? Okay. Okay. Sorry. So yes, I was talking about this particular stage. Yes, so they give you an agent before present, like BP. Now this is because, now why 1950 for BP and not the present year, the current year? Now this is one of the reasons for this is because of the nuclear weapons testing. So what happened is, during 1950s and 60s, we had a lot of nuclear weapons tested around the world because of Cold War after 1960s and stuff. So that dramatically altered the atmospheric carbon 14 ratio, and hence we needed a fixed reference point before that contamination. And hence the scholars, they decided on 1950, which was also the year this entire process it started, like it began being used in archaeology. Then also it is important to keep in mind that the radiocarbon date, it tells you the when this particular organism died or when it stopped exchanging carbon with the atmosphere. It does not tell you when the object was made or when it was deposited or when a cultural event around this object happened. It just tells you the moment, like not the moment, the range of age where this organism died or stopped exchanging this carbon. So the the gap between the processes I told you about, like the initial making or deposition or working of this object, for example charcoal, and the date it was buried or died, are completely different phenomena. So these could be centuries apart. For example, you could have a tree log which you cut for making something, but then you did not make something out of it for decades, and then you made something out of it after example 50 years. So now this will not tell you the radiocarbon date. This will not tell you exactly when you made something out of it, right? It will tell you the process of carbon exchange will stop as soon as you cut the tree. So there will be a gap of 50 years over there as well. So we must be very careful when we're working with radiocarbon dates as well. And we also must think about the sample's life history from the moment it formed to the moment it was excavated. Then this is a pictorial representation of what I told you about. If you look at this, like these are the cosmic rays, and then they're being converted into carbon 14. Now this carbon 14 is being converted into carbon dioxide, being absorbed by the plants, which are eaten by animals, which this animal is again eaten by another animal, and then this animal died, and now we have this process of beta decay, like carbon 14 being converted back into nitrogen 14. And then now, if we look at this curve over here, now note that this is not a flat line. Okay, so this is a decay curve. So this is not a flat line. This drops steeply for the initial years, like the first half life, and then it's a little flat, like it becomes a little flat, like close to flat as we move on, right? So after first half life, that is after 5730 years, we'll be left with half of whatever carbon was there in one organism. And then again after another half life, that is after another 5730 years, that is total 11,460 years, we'll be left with a quarter of the total, like of 100, right? And as we move on and on and on, uh, we'll be left with half and half and half and half and half of whatever this value will be, the recent value will be. Now we can measure it up to 10 half lives. Now why 10 half lives? Because after 10 half lives, it becomes the remaining carbon, it becomes so little that it is practically not reliable, and also not, very difficult to measure it. And if we convert these 10 half lives, it will come out to be around 50,000 years. So that is the practical limit for radiocarbon dating as well. Now with AMS, accelerated mass spectrometry, which I mentioned in the previous slide, we can push this a little further. And the best part about that method is that it requires very little amount of sample, while radiocarbon dating requires about 1 to 10 grams of sample. Now an important part, which is like, if we are looking at radiocarbon dating, we must look at calibration as well. So labs, we talked about labs giving us this raw date, right? So date we get this date not in like proper terms of BCE and AD, but in BP, before present, which is before 1950. So we get these raw dates from these labs. For example, we get 3500 plus minus 40 BP. So this will be our raw date. But then this is not the calendar year, right? We need to convert it into one. to understand it better. So we need to convert it into before Christ, like before Common Era or Common Era, BCE or or CE, right? So for this conversion, we need to calibrate it. Now, why is this calibration even required? Why is it even necessary? So the atmosphere does not actually produce carbon 14 as a constant rate, while it is a constant process and it keeps on happening, like it's still happening in the atmosphere, right? But then it is not it does not produce this carbon 14 at a constant rate only. This rate, it changes because of several factors. One of these factors is the sun's activity, which keeps on fluctuating. Other factor might be the geomagnetic field strength of the earth, which changes over millennia. So we have these factors which keeps on changing, and hence the rate of carbon 14 production also changes. And also we must note that carbon 14 to carbon 12 ratio is not constant in the atmosphere. Like we talked about that nuclear thing, right, in 1950s, 1960s, right? So the ratio, it changes, it does not remain constant for every year, and hence we need to calibrate it. So, for example, for some centuries, the organism would have maybe absorbed more of carbon 14, and maybe in another some centuries, it would have absorbed very less amount of carbon 14. So, it actually depends, right? And hence, we calibrate it to have this figure with us, which is reliable. So for this, to understand this, we have a calibration curve as well. Now scientists, what they did was they constructed a calibration curve based on the carbon content of material whose actual date was known, right? Like they knew the independent date of these material. Now this material was primarily the tree rings. So what happens with the tree ring is, per year, like every year, they add on one ring, right? And that is an actual calendar years. So they tested it against those tree rings. So if I talk about like specifically, they used Irish oak and also German oak. Now this sequence of German oak, it stretched back to about 14,000 years, and it also, and because radiocarbon gives us a date of 50,000, right? And these tree rings were giving us a date of 14,000. So we needed to stretch it further. To extend it further, they also relied on coral dates, coral material samples, and cave formations, the paleothems. They gave us, they are very reliable for dating, and also deep sea sediments. So they relied on all of this to make this calibration curve. Now, I talked about German oak and Irish oak, right? And not Indian oak, like Indian tree, right? So in India, we do not yet have a dedicated regional calibration data set, which is one of the reasons that, like refining, like we are still refining that. We're still refining the South Asian carbon dates, like radiocarbon chronologies, and this is an active research problem which some scholars, Indian scholars from BSIP, are also working on actively to create this Indian data set, our own calibration curve, because again, this rate is not constant, and also it depends on the region. I'll be talking about this regional aspect later on as well. So the current standard, the international standard, we'll be talking about that now, because we do not have an Indian standard, right? So international standard, and everyone follows the international standard. So the Indian international standard is the INTCAL 20, which was published in 2020. Okay, so this calibrated output is always in probability distribution. So it does not give us a single year, but a range. So like this, right? So we won't get a single year like 1820 BCE. We'll get a range of it from 1880 to 1760 BCE, for example. And what is the confidence percentage for this? 95.4%. So that typically means that we are reporting it at 95.4% confidence that the age range for this particular object is 1880 to 1760 calibrated BCE. So over here also you can see CAL, right? So this is how reports and scientific papers, they mention calibrated. dates. So you guys must have read some scientific papers, journal articles, reports, etc., right? So you guys would have maybe also noticed that sometimes there's a big table given over there, where they've written sample number this, then age and BP. They have a range of dates for that, and then age in calibrated BC, right? So we have this. So good science papers, they always mention the uncalibrated, that is BP age, and the calibrated, that is BCE or CE age. Now coming to the actual measuring part. So how do we actually measure carbon 14? Now we get this sample at a lab, right? Now we have two methods of counting or of calculating the carbon 14. Now one is beta counting and the other is accelerated mass spectrometry, AMS. So beta counting is the original, the OG method, which was used by Libby, which was developed by Libby in 1950. In this, we get this sample and we chemically convert it into gas or liquid, placed in a detector, and then the machine counts the electrons emitted by the carbon 14 atoms, decaying over several days. So we just create this, we create the conditions for this decay. So we measure the decay, right? So we recreate this process of decaying, and we measure it through this process. Now this requires about 1 to 10 grams of sample, and it takes long time, like it takes several days. And on the other hand, when we have AMS, that is accelerated mass spectrometry, which we talked about in the earlier slides, it came into being from 1977. Now the good aspect about this is that we don't need to recreate this entire process, and also we don't need this large amount of data, of a sample, right? Sample size. So the sample is very small, like it can be done on a single grain as well, single seed as well. So we just need 1 milligram or even less. So that tiny amount of sample can tell us a lot, right? So over here for beta counting, a huge amount of sample is being destructed, right? 10 grams and even more, right? So it is being destructed because it is being converted into or liquid, right? It is being destructed. So so in that way it is very useful. Also also it is very fast, so you can get the results within hours or only a few days at max. So you get that result a lot faster and also with a lot of precision. So if we talk about beta counting, it gives us a range, and the age range, it could be a huge gap between those, right? So for plus minus that we talked about earlier, so it could be plus minus 150, it could be plus minus 200. But for AMS, this precision rate is only about plus minus 20 to 30 in good conditions, or maybe 40, etc., right? Um, so over here, if but then yes, again it also has its own limitations. It is very expensive. When I was searching about it, I'm not sure what the actual cost is, but then when I was searching about it, it is around $2 million. So it is expensive, and it also requires specialist technicians. So a lot of Indian scholars, they also rely on foreign labs for AMS. Not that we don't have them in India, we have them, right? We have that at Ahmedabad, we have that at Lucknow, and scholars do send their samples over there as well. But then a lot of samples are also sent abroad because of specialist technicians that are there in those labs. And and also it can also magnify the contamination problem, because we have such a small amount of sample. Any tiny or minute contamination, like very little amount of contamination, would also be magnified over here because the sample is also very small, right? Now if we talk about Ahmedabad, right? So they and also BSIP, they both have their own AMS machines. And BSIP also has their automated graphitization equipment system. So, which I was talking about, it was established in 2017 or 18. So, what happens in that particular machine is they prepare the samples in that machine. Now, this machine, like this introduction of this machine in Indian context, it has been very, very helpful because now scholars can send their material with a data of pre-treatment to the Indian labs. Earlier a lot of samples were sent abroad. Even now they do. For Killary, they sent their samples to Beta Analytics in USA, which is a very renowned laboratory, honestly speaking. And also, like sending the samples abroad, I'm not saying that it is wrong, okay? So it is normal, it is a normal international practice to send these samples abroad. And also it does not reflect that it does not reflect any lack of Indian capability, okay? It simply reflects the global demand for AMS dates, and also specific expertise being required for different sample types. Now what matters is that the laboratory to which we are sending our samples is accredited, and it uses internationally recognized standards. And also the reports in and its results are presented with full documentation of pre-treatment and the measurement uncertainty. So that is again a good science practice to always mention the uncertainties and also the pre-treatment, like how it was done. Then we have talked a bit about carbon 14 dating till now, and now we should also look at the limitations, right? It is not that it is a gold standard and then it's all good. It's not like it's all pink picture, right? So it has its own set of limitations, like every science does. Every method in science has its own limitations. So the first limitation or challenge is contamination. So any foreign carbon introduced in the sample will ruin your date. So any modern thread or roots or water or anything like that, it will like anything at all, it will tamper with your results significantly. And hence it is very important that every sample that you are collecting is done with accuracy, is done with precision, and is done properly. Now they should be stored properly to be transported to the lab as well. Like while you're collecting it as an archaeologist, it is your responsibility to collect these samples accurately, right? To label them accurately as well, and also to transport them to the lab properly. And also, while you're sending a sample to a lab, it should also go under proper pre-treatment, which is like it is very important for the sample to go under pre-treatment. Bizarre dates, like it could be too old or maybe too new, right? It will give you dates which don't make sense. Okay, so in practice, if we talk about what an archaeologist should do in such cases, like while collecting the sample for radiocarbon dating, if you're collecting a charcoal, it should be properly wrapped, like in clean aluminium foil. You should collect it in a clean aluminium foil and never directly in a plastic bag, because that can harbor organic contamination. And also it should be labeled, like after you collect it in a aluminium foil, wrap it properly, and then keep that aluminium foil in another bag, right? And label that properly. And when I'm saying labeling properly, I don't just mean that mention the name of the lab you're sending it to and mention that this is charcoal. I mean labeling it with context information, the layer and everything. Like you should label everything precisely, and also maintain a strict chain of custody from the excavation to laboratory. It should not be hanging around anywhere in some corner at your site. It should be handled properly and sent properly to the laboratory. Once the like if we're collecting it, and if we don't send just one sample, okay, we send a lot. So if for example if you send a few samples and one out of them is contaminated, mm, the entire sequence, like the entire data will face these reliability issues then. Like if the dates are not consistent, if they're misleading, the entire interpretation could be considered maybe rigged or maybe not accepted by scholars. The second limitation would be the reservoir effect. So we need a little extra care over here. Ocean, right? So okay, so oceans contain old carbon. Deep oceans, they contain old carbon. So in an ocean, the deep water, it circulates for centuries before it finally comes at the surface, right? So in that case, they they also, the marine animals, the organic beings in that deep water, they also appear older than they actually are in radiocarbon dates. And this, when I'm saying appearing older, I mean older than they actually are, like on an average 400 years. And this is called the marine reservoir effect. For Indian context, if we're talking about Indian context particularly, I believe the samples from the coast of Gujarat, Konkan, Tamil Nadu, Orissa, etc., the marine shells also, these samples should be treated properly, and they need reservoir corrections as well. So what they do is, like, as I said, for calibration, right, they have these correction rates. So for marine samples, they have a different calibration rate or correction rate, and for terrestrial ones, they have a different correction rate. So the exact correction value over here, it's not that it will be 400 only. It varies based on the region and also the period. So like, based on the region you're collecting your sample from, the same data will be corrected. And also, like we have marine reservoir effect, we also have a fresh water reservoir effect. If we talk about humans, so for human bones, okay, human bones can also be dated through C14 dating. And what happens is, if a human or a population consumes a lot of freshwater fish on a daily basis, the dates may appear older for those particular bones than the ones who are not the populations who are not consuming freshwater fishes that often. So Indian Ocean also has its own regional reservoir variation, and the correction factor for, say, for Gulf of Khambat, for example, is not the same as it would be for Coromandel Coast. They will be different. So this is an area of, again, ongoing research, and it is one reason why some published marine shell dates from the Harappan coast, they carry a wider uncertainty ranges than comparable terrestrial samples. So if you like, I would recommend you going and reading about this a little. Like, if you look at some of the reports from for Harappan period, like, you can take any period that you like. So they also have this dating for marine shells, and the uncertainty over there is actually huge. I don't remember the dates exactly right now, but then I can definitely share the papers. Then again, we have the third limitation. So okay, also before this limitation, there's also another one, the old wood one. Okay, so there's this old wood problem. Now look at the trees. Okay, one tree might grow for about 200 years before falling. Okay, now after it is cut to be used somewhere, it may have been kept for decades, right? So you may keep it for a long period, but then the carbon dates for this particular tree that we're talking about, they will tell us when the tree died, and not when it got associated with any cultural phenomena, or not when it was being used by the humans, right? So this is why archaeologists, they always prefer the short-lived material like seeds or charred grains, etc. So these are always preferred by the archaeologists when they're working with radiocarbon dates. Then we have the plateau problem.
Aku maklang marku
Can you mute yourself?
dulu eh
Okay. Someone is singing, I think. Please mute yourself. Please don't do this. Okay, so we're talking about the plateau. So we were talking about the plateau problem, and if we like, there are some periods in calibration curve as well where the atmospheric can you
ಆರುತ್ ನೀ ಒಟ್ಟ ಅನ್ನಕ್ ಹೋಗ್ಬಿಡ ಅಲ್ಲ ಆರು ಬಿಡ
do
ತಲೆಯಿಂದ
you have a question?
ಮಳೆ ಬರ್ತೀನ ಒಂದ್ ಸರ್ವ ಒಟ್ಟ
I don't think I have that. Sir will have to mute him. Sir, can you please mute him? I'm not the admin. Okay, thank you. Okay, so we were talking about the plateau problem. Okay, so what happens is when we're looking at calibration curves, so these curves are not always perfect. Okay, so they also have these plateaus, like plateaus type of feature in them. Now what happens is atmospheric carbon 14 ratio, it changes for centuries. Like it cannot remain constant, like we said, like it does not remain constant, right? So we have these plateaus, which is a single raw carbon date maps an extremely wider range of possible calendar dates, and sometimes it can go on for centuries, like 2 centuries or so. And this is a major problem, at least in European case, which I came across. So in European case, particularly in Iron Age, for their Iron Age, which is about 800 to 400 BCE, they have this plateau problem, like the plateaux is what is it's called. So I would like you all to search about this problem in Indian archaeology as well. Like it will be interesting. I haven't searched too much on it myself. I'll also go and search about it for Indian context, and I'd like for you all of you to also go back and search for it after this class. And this is an interesting aspect for radiocarbon dating, a limitation that is not really common, but then again, one big limitation. Then again, we have the bomb pulse carbon as well, which is the reason we use 1950 as the standard rate. Like I told you about these nuclear weapons being tested during the time of Cold War, right? And because of that, it altered the carbon 14 ratio in the atmosphere, and hence we use 1950 as the standard and not the later on dates and not the like present date. And also we needed a standard, so it is better to have one standard for all the samples. So we have that 1950. Now coming to our last slide, which is like we have all have this basic understanding, I believe, of carbon 14 dating, its mechanism, and also some of its limitations. So we must also know about what we can actually date, right? We as archaeologists, we should know about what material is dateable and what is not directly dateable by a particular technique, dating technique that we're using. Now one thing is, only and only organic material can be dated through this method. Okay, so charcoal is one of the most commonly plateaus. Okay, so I'll explain it. Now what this thing is, in the calibration curve, in the decay curve, sorry, in the decay curve that I showed you over here, right? So in this curve, and similarly we also have a calibration curve, right? So in that calibration curve, we don't always have this particular shape. We also have sometimes, this is the decay curve, but then the calibration one, we also have this plateau kind of thing, right? So it will start, and then it will be a flat plateau, and then go back to its constant rate, or similar to constant rate, the changing rate. So now this straight line, that is created in the curve is basically because of these fluctuations in carbon 14 in the atmosphere. Okay, so what this creates is it can go on for about like centuries, 2 centuries, 3 centuries, and so. And this becomes a major issue when we are dating the sites, because it will not give us the right rate. It will tell us a fluctuated dates. It won't be like giving us dates which are too haywire or too bizarre, but then it will give us like major gaps between the dates, which will be an issue in research field. So that is what I was talking about when I mentioned plateau. And you will have to look for Indian examples in that. I'll also share one if I come across one from the Indian context. I'll also go and study for Indian one. For European one, I did for Iron Age sites. So they are always skeptical of their radiocarbon dates for Iron Age sites in European context because of this problem. Yes, so coming back to the material, so charcoal is one of the most commonly dated sample in Indian context and in radiocarbon dating uh, generally, because it also gives us a very precise and reliable date. Then we also have burnt wood. We also have all of these organic materials, seeds and plant remains, bones, shells, and then textile, organic sediments, leather, antler, ivory, everything that is organic in this entire world is datable through radiocarbon dating. So if we're talking about particularly about seeds and uh, charred grains, they provide exceptionally reliable dates for reports. And also if we're talking about bones, like I've written via collagen, right? So why is that like this? So what happens is, to date the bones via radiocarbon dating, we don't actually date, like we actually focus more on collagen, that is the protein inside the bone. So we date that particular part of the bone, the collagen. However, in Indian context, if we talk about this, so it doesn't really survive in most of Indian archaeological context, and the reason is the environment. So this collagen, it degrades rapidly because of humid conditions, and hence in peninsular India and also in Gangetic plains, uh, these environments, they don't prove to be very suitable for the prevention of uh, bone protein to be preserved, to be dated, right? For textile, any piece of fragment, we can date any piece of fragment, but then again, if it survives, so which is a little rare in Indian context for early history, or prehistory or Mesolithic period and stuff, right? For and Yes, also talking about seeds and plant remains, which are again short-lived samples. The charred grains, for example, they are always preferred over the long-lived ones. So if you've been to a site, to an excavation, whenever we find any charred grain, we're all very excited, and we're all very excited to uh, carefully collect it for sampling, because that is something that would give us a very reliable date. And also something that actually, like, it is like a gold. Okay, it is like gold on an excavation site if we're going for radiocarbon dating. Okay, so it is it gives us a date which is very close to the occupation event, because it doesn't it would have been it's not that you could have protected that grain for like a seed which is not charred. You could have protected it for centuries, right, or decades. You grow grains every year, right? So, and if you're cooking it, if you're using it, so it gives us a very reliable and close to actual occupation or habitation occupation event date. And also a piece of structural timber from a building, might have been a wood from a tree that grew and fell maybe 2 to 3 decades or centuries ago, which we talked about, the old wood problem. So hence wood is again a little skeptical for some scholars when they're looking at the report. Now when you have a choice, like if you do, if you're an archaeologist, and in the future if you get this choice, always and always prefer the seeds. Like if you have charred grains or seeds from your site, do send them for sampling. And also now we've talked about doable materials, or like dateable ones, the doable things that can be done with dating. Now we'll talk about the ones which cannot be directly dated. So we cannot date the stone tools, they're not organic. We cannot date the pottery directly, however we can date the organic residue inside the pottery. So we sometimes find charred grains inside potsherds, so that can be dated, but not the pottery itself, not the stone tools itself. Then we cannot date metals, we cannot date any inorganic sediment, we cannot date any glass or obsidian material as well. So as I said, we need organic material for this particular dating method, dating technique. For these we have other methods of dating, scientific methods of dating. We have, for instance, TL dating, thermoluminescence dating. We have optically stimulated luminescence, or OSL dating. Now these are widely used in Indian archaeology, and these methods, they tell us about the last time this material was, whatever material we are sending for sampling, this material was heated or exposed to sunlight, to light. So um, um, in terms of pottery, for example, if we are sending it for TL dating, it will tell us the last firing event, last time it was fired. Hmm. So BSIP Lucknow and also PRL Ahmedabad, they also have a lot of, and also other lot of Indian institutions. They also have TL and OSL facilities, and uh, like make it also. I would recommend, like if you're going for dating, it's not always that you have to rely on only one dating method. You can go on with several dating methods, also depends on the funding that you're getting for a particular research. So it's always better to go to build a multi-method chronology that combines carbon dating and other dating methods like the luminescence dates, etc., for the same site. It 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 gives you a more reliable report or data for your particular research. And then finally, some of the best practices that we all must adhere to while we're working at a site as archaeologists and dealing with radiocarbon dating. So like one is preferring of shots, like I said, to avoid the old wood effect. Then also sampling from a very secure primary context, and very carefully in the aluminium foil if we're collecting charcoal samples. And also every material has a different like it's not that you can treat every material similarly. So you have different treatments for every material, different ways of protecting every material while sampling methods are also different for materials, so you'll have to learn that, you'll have to study about that as well. Hmm. Then also doing multiple like using multiple dating techniques on a particular context, at a particular site, is definitely one of the best methods. It also gives you this transparency, so you're not relying on only one date, but you're also working with other dates. So if they're giving you similar range or same date, that means that you're doing it right. It gives you the confidence like it gave to Williard Libby.